Welcome to the Colombian gene video, which gives us a gentle introduction to simple applied chicken genetics. We will learn about the inheritance of a simple dominant gene, but also get a better understanding of genetic ratios and expected outcomes from breeding experiments. The Columbian gene is an autosomal dominant gene that restricts black pigment to the extremes of the bird. These are typically the hackle feathers, the end of the tail, and sometimes black in wing feathers. It should be noted that this pattern does not work on the extended black genotype. Birds with extended black stay black. As we can see in this Brahma, the Columbian gene also doesn't affect the pheomelanin pigments. The gene is considered to be fully dominant, but there can be minor variation in females where some black pigment may appear in other areas, especially across the back. In combination with some genotypes, Columbian may restrict black from the tail, leaving black just in the hackle feathers. Since the Columbian allele is fully dominant over the wild-type allele, it is ideal for demonstrating simple inheritance. First of all, let us consider what happens if we cross two Columbian birds together. The first problem we have is knowing whether those Columbian birds are homozygous or heterozygous for the Columbian allele. We can immediately see that there are three possible crosses depending on the genotypes of the birds that we are crossing. In the first case, both birds are homozygous for the Columbian allele, so the chicks could only possibly be homozygous for the Columbian allele as well. In the second case, one of the parents is heterozygous. They produce a 1-to-1 ratio of Columbian and wild-type alleles, so that half the chicks are also heterozygous at the Columbian loci. Before we go on to look at a cross between heterozygous birds, this is a good place to consider what is meant by the ratios we see from crosses. The ratios show what we expect to see on average from a large number of observations. An easy example to understand is the sex of chicks on hatching. We expect approximately half of them to be girls and half of them to be boys. The more chicks we hatch, the more likely it is that there will be a 1-to-1 ratio of girls to boys. However, for any particular hatch, there is a certain amount of chance that the ratio will be skewed either towards boys or towards girls. I can illustrate this with something that happened to me about 4 or 5 years ago when I hatched 23 eggs one spring. Just by chance, 17 of my lovely birds grew up into cockerels. It can be worked out using a binomial distribution if anybody wants to do it, the probability of a particular number of girls or boys in any particular hatch. For a hatch of 23, there's a 59.5% chance that 10 to 14 of them will be boys. You can see there is just a small percentage chance of there being 17 or more boys in the same hatch. It's a small chance, but not zero, so someone eventually will probably have similar results to me, given how many chicken breeders there are in the world. I've not known anyone else have such an extreme result, and nor have I, either before or since. Most people will get probably between about 10 and 14 boys if they hatch 23 chicks. However, I just wanted to show you that it's possible to get other results. Anyway, back to our third possible cross, which is between two birds that are heterozygous for Columbian. The cross between two heterozygous birds is called a monohybrid cross, and we use the Punnett square to find out what results we can expect. In this case, both birds have gametes for both the Columbian allele and for the wild-type allele, and these are represented along the top and the left side of our Punnett square. Then by working along and down, we can easily work out what the phenotypes and genotypes of the chicks are likely to be. We can see that there is a characteristic 1:2:1 ratio of genotypes and a 3:1 ratio of phenotypes. So that on average, we could expect 3 Colombian chicks to every wild-type chick. We can also use genetic ratios the other way around to make inferences about parent birds from their chicks. If, on crossing 2 Colombian birds, some of the chicks are wild-type without Columbian patterning, then we know both parents are heterozygous for Columbian. On the other hand, if we only get Columbian pattern chicks, then we can assume that either at least one parent is homozygous or that we have too small a sample size to be certain either way. In this case, we would either need further information about the parents or we'd need to make further crosses to decide on the parental genotypes. Let us consider briefly now crossing Columbian and wild-type birds. The genotypes of the Columbian could be homozygous or heterozygous, and the wild type will be homozygous for the wild type allele. In the case where both birds are homozygous for the alternative alleles, all the chicks will be heterozygous for Columbian. If one of the parents is heterozygous for Columbian, then we could expect a 1:1 ratio of wild type to heterozygous Columbian chick. From this video, remember that Columbian restricts black to the hackle, tail and wing feathers. It is a dominant gene and simply inherited. Also remember that genetic ratios are the expected average likely outcome over a lot of observations. The smaller the number of observations you have, the greater is the chance of there being a deviation from the expected ratio. Thank you very much for watching this video. If you have found it useful, please like, comment below and share it with your friends or on social media. Subscribe so that you're notified of chicken genetics videos as they come out. I hope to see you very soon.